I think we have a fellow Gonzagite.
Yeah, cool.
Then did his PhD at the UW, through the UW MCV program, right? Although he did his training at the Fred Hutch. And for people who go through the PhD route, he's done something that's a little bit unconventional or off the traditional road, and it's kind of stuff he's going to end up talking about today. So he's got an awesome talk to give us, so I'm not going to spend much more time. I'll turn it over to Colin.
Hello, everybody. Great to be here. I really love, especially somebody that's so young in their kind of drug development career, I love talking to young kids about drug discovery because I didn't learn any of this stuff as an undergrad, and believe it or not, I didn't learn any of this stuff as a graduate student because one of the problems with graduate training is that it's often so focused sort of on understanding basic principles and problem solving that you actually finish your PhD or whatever graduate work that you've done, and you really still are not equipped to go out into industry, to go out into sort of like a startup biotech company, for example, and really do anything unless you have a very specific skill set that you develop during your graduate work. We really do a bad job of training graduate students for different career paths outside of the traditional academic career path, where you would go on and do a postdoctoral fellowship in a lab where you're very interested in sort of carving out your own niche within science, and then hopefully being very successful during your postdoc and being able to compete for faculty positions. But that's very much how graduate school is kind of set up for a lot of scientists. So for those of you who really love science and who either want to go into medicine or into research, it's good to be thinking about these things and seek out the types of training opportunities that you think you're going to need to be successful at the later stages of your career, because so many people get done with their graduate studies and feel kind of helpless or hopeless, meaning that they don't really have any of the things to compete for the types of jobs that are out there. So the earlier you guys can start thinking about this in your careers, you will be, light years ahead of most of your peers in terms of competing for positions out there. So like Brett said, I took sort of a non-traditional approach to this. I knew I didn't want to get onto the sort of traditional academic track. So I really focused during my graduate years on doing work that was applicable to industry so that I could go out and actually compete for a job, hopefully right out of graduate school, which is something that very rarely happens. As it turns out, I ended up foregoing a job at a little startup biotech company to stay at the Fred Hutch and work with a PI named Jim Olson. And hopefully, did you guys watch the little YouTube video? Was that the TED Talk that Brett linked to you guys? Or?
It looked like a TED Talk to me.
Yeah. And so I'm going to talk a little bit about some of the stuff that he talked about in terms of the development of this tumor paint molecule and where we're taking this research at this point. And then hopefully, if I have some time at the end of the talk, I can kind of get up on my soapbox again and talk to you a little bit about drug discovery as a whole, because it's actually a really cool industry and it's a really cool place for people who are trained in science to go. But that's sometimes lost on students because it's almost an impenetrable career that you don't know anything about until you actually get onto the ground floor there and are lucky enough to start working there and learn as you're there. So hopefully this gets you guys thinking about this. So just to root everybody, our lab and everything we do in the lab is based on these kids. These are all patients of Jim Olson. All of these kids, beautiful, beautiful kids, had rare pediatric brain cancers, and every single one of them died from their pediatric brain cancers. These are very rare diseases, so there's sort of no incentive for large drug companies to do research against these types of cancers because the market is just too small, and it doesn't justify the huge amounts of R&D costs that go into actually bringing a drug into the clinic. And this is what Jim sees every day in the clinic, are these kids. And their diseases were such that there was essentially no hope for new therapies. These are either kids that have had two brain tumors that could not be resected, brain tumors that don't respond to kind of what I call the carpet bombing techniques that we use to treat cancer nowadays, that is chemotherapy. Really no hope at all. And so these are the kids that basically drive our lab, and it's their families who fundraise for our lab, and it's it's the families who basically go out there and beat the streets and have bake sales and help raise money for the lab that keeps this research going. And I'll tell you this, the families of kids like this are the families that actually made Tumor Paint a reality. We didn't have R01 grants that brought this molecule into the clinic. This was all basically done through philanthropic means, mostly through gracious patient families. So we owe a lot to these little guys. And as you see on my first slide, I have this moniker, Project Violet, in memory of these patients, Jim started Project Violet. And Project Violet basically is a front porch for our research so that new families can come learn about our research. They can fundraise under the Project Violet umbrella. And it allows us to basically educate people on the type of work that we're doing at the Fred Hutch. So, and it's really caught on. We've gotten a lot of really great press surrounding Project Violet, all the way from Seattle Times to National Geographic, BBC. So there's a lot of information out there on the stuff that we're doing. So if you're interested, you can find a lot of that. And I think it's also a testament to what we've built here. This is not traditional drug discovery that you would see at a big biotech company like an Amgen or Genentech. This is essentially drug discovery that's being funded by patient families, and it's being done at the Fred Hutch. And we're focusing on kids with rare cancers. So this is the first part of this talk. I'm just going to kind of take you through Tumor Paint and where that is in the process of drug discovery. And I'm going to talk to you about what we're doing to follow up on the Tumor Paint story. So this is something that Jim sees all too often in clinic. This is a scan of a brain, and you can see that there's a big, massive brain tumor here. This is what the surgeon sees. He gets an MRI, and from this he has to decide what the margins are to resect this particular type of tumor. The first line of defense against solid tumor diseases like this is surgical resection. And it's very clearly been shown that the more of the tumor that you get out and the more of the healthy tissue that you leave behind, the better those patients do. Pretty simple to think about it that way. Unfortunately, this is what a surgeon sees in the operating room. Can you tell where the tumor is and where the brain is? This is a huge problem. And especially for these kids who are undergoing surgery at such a young age, if you take out just a little bit too much of the healthy tissue, these kids have major cognitive issues as they grow up if they actually survive the cancer that they're dealing with. And the tools that the surgeons have in the operating room this time are pretty primitive. It's their fingers and their hands and some electrodes that they can use to try and tell the difference between a brain cell and a cancer cell. And ultimately, this is what happens in surgery rooms all across the United States, is that a Harvard-trained neurosurgeon in this case spent 18 hours in the operating room, and when they come back and scan this patient, you can see there's a thumb-sized piece of cancer left in this patient. So this is sort of what inspired Tumor Paint is, can we develop a molecule that actually guides the surgeons to actually make surgical resections more productive? This is the Tumor Paint molecule. This was developed in Jim Olson's lab. A lot of this initial work was done before I even joined the lab. And it's made up of two components. A very small protein molecule. This is called a Naughton protein, which I'll talk about a little bit later. And this protein actually comes from the venom of the deathstalker scorpion. You've been learning about proteins in this class. This is a very unique class of proteins, and I'll get into this class a little bit later. This protein is covalently linked to a near-infrared dye called ICG. And this is a dye that's been used in the clinic for like 50 years. So it's very safe. And we know basically its toxicity profiles, everything like that. It's been used forever. And this conjugate basically makes a drug that glows under near-infrared light. So you can see this is a picture of Blaze 100, which is the clinical candidate that's in clinical trials right now. And when you illuminate it with a laser, it glows bright. So you can see exactly what this molecule is intended to do. And it's got to be one of the coolest looking drugs I've ever seen, for sure. So what Tumor Paint does is it basically has an affinity for cancer cells. So when you inject this drug either into the tail vein of a mouse or into the bloodstream of a human, it drags this little molecular flashlight to the cancer cells and illuminates them. What I'm showing you here, yeah, you can see this, okay, are basically two panels of a mouse brain. And this is being illuminated by near-infrared light. And this is the H&E staining. And so if you look over here into a healthy brain, you see the Tumor Paint molecule, with the exception of a few areas, these are like the ventricles of the brain, it doesn't really light up the brain at all. And this corresponds to the H&E staining. However, in this cancerous brain, this is a mouse that spontaneously develops a type of pediatric brain cancer called medulloblastoma. So at a few weeks of age, these mice just spontaneously start growing these tumors. You can see that the Tumor Paint molecule, almost on a cellular level, is lighting up this developing tumor in the brain. And this is shown here in the H&E staining. It's kind of hard for you guys to see because the resolution is not that great. But if you could see, you'd see there's this just massive amount of cancer cells right here. And so this was really the first discovery that kind of led Tumor Paint to start being brought into the clinic is the fact that this thing was lighting up cancer cells. And I'm going to kind of skip through this process kind of quickly, but based on the early data from mouse models of cancer, this molecule was taken into a variety of different mice that had a variety of different types of solid tumors, brain cancers, colon cancers, breast cancers, and time again, this molecule was lighting up these cancers inside the mice. And so they started a company called Blaze Bioscience to commercialize this product. And Blaze basically took this molecule and leveraged all of that mouse data and said, okay, we need to get into higher animals, into people as quickly as possible. And so that's exactly what they did. This image is from a dog. This is done with people who voluntarily bring their dog into the Wazoo Vet Clinic. This is a collaboration with the Wazoo Vet Clinic. And oftentimes, in the case of a lot of different types of canine cancers, you'll either amputate the leg that has the cancer, or you'll just let the dog die because you don't want to pay for the cancer surgery. But these are parents who really love their pets and wanted to see if they could actually cure their pets. And so they took the Tumor Paint molecule into this dog model. This is a mammary tumor in a dog. And you can see, this is the near-infrared overlay, that the Tumor Paint is really beautifully lighting up this cancer mass. One of the things I hope you guys can appreciate is that just by looking at this picture, it's really tough to tell where the cancer is and where the human tissue is. This is clearly the tumor, but Tumor Paint also lights up a bunch of other spots here. These are what we call skip lesions. And these are things that if you were to resect this tumor, the surgeon might miss. And this is something that happens in surgery rooms across the nation where there's cancer that's left behind because the physician doesn't know exactly where it is or where it isn't. So this is a beautiful thing that it was translating from mice into canines. So you can see we're kind of moving up the animal chain here. And then very quickly they started taking this into human clinical trials. And I'm going to show you, courtesy of Blaze Bioscience, some of the first data from clinical patients. This is Tumor Paint in humans. So this is a resection of a breast cancer in this case, and you can see that they've taken a very large chunk of tissue out. It's, again, very hard to tell where the tumor is. And then this is the near-infrared overlay. So you'll kind of notice that these images kind of look a little weird, but it's just because they're basically superimposing what you would see under near-infrared light on top of the white light images here. And you can see very clearly that Tumor Paint is lighting up this tumor. And you can see here that the surgeon in this case has probably taken a really margin. So the way this is working in clinical trials right now is that the surgeon can't actually use Tumor Paint to guide the surgery, but they can go back afterwards when the tumor is actually set down to pathology, and they take a look at the cross sections of the tumor, and this is how they determine if a margin is clear of cancer cells or not. And they can say margins are clear by H&E, and then they can go back subsequently and look at the Tumor Paint data and say margins are clear by Tumor Paint. And that's basically how this clinical trial is running at this point. The surgeons can't actually use it because it's a clinical trial to make surgical decisions, but we can retrospectively go back and see, is the Tumor Paint molecule telling the surgeon what we want to tell the surgeon. And then to bring this whole story kind of full circle back to the patients that Tumor Paint was actually developed for, this is an example of a pediatric patient that was in the Tumor Paint trial. And again, you can see here's the preoperative MRI with a very large tumor mass. And then this is what the surgeon actually sees in the operating room. And you can see that there are areas of the brain here that are lighting up very bright with the Tumor Paint molecule. We can't really get specifics because of the restrictions of clinical trials, but we have heard stories of the physicians who will be doing brain surgery. They'll finish up with their brain surgery, and then they'll turn on the near-infrared light, and they will see cancer that's been left behind. And they can go back in and take that cancer out without having to stitch up the patient and go back later. So it's already having an impact on patients, and that's a pretty remarkable thing. This molecule has come from the lab and into clinical trials at breakneck speeds and with dollar amounts that you can't even imagine. So I think it's a really remarkable success story, but it's really only part of the thing that we're trying to solve in Jim's lab, right? We can bring light to a tumor, but now can we actually develop therapeutics? Can we leverage the properties of these little small proteins that we found from the deathstalker scorpion and actually use them as targeting agents? Can we now go from bringing light into the cancer into bringing cytotoxic reagents into the cancer? So, for example, making chemotherapy much more specific. So this is just a summary of some of the Blaze clinical trial stuff. It's been in over 60 patients, which is just a really remarkable thing. The other remarkable thing about this is that of these patients, A subset of them are pediatric patients. One thing that people don't appreciate about drug discovery is that most drug trials are run in adults, and they're almost never run in children. And all the drugs that we have that are used in children are basically used off-label because we think that they will work in the children. And this was one of Jim's promises to his patients, was that we're going to run this clinical trial, we're going to run it in kids too, to make sure that it works in kids. So it's a really remarkable story. We continue to get data from the clinic almost daily about this molecule, and we're really excited to see where it goes. So I just wanted to quickly tell you that story of Tumor Paint, because it really kind of sets the stage for the work that we're doing now in the lab. And I'm going to talk to you about what we call the OpType program, which is essentially taking these small proteins from nature and weaponizing them in a way that we actually leverage their sort of natural properties. We often refer to this as drug discovery, kind of inspired by nature. The fact is, nature's done a lot of drug discovery on its own, things that we as drug discoverers have never even thought of. And so can we leverage these properties to develop next generation drugs? And at the heart of this is trying to drug essentially what we call undruggable targets. So I love to show this slide because it's a really good example of what it is that we try to do in drug discovery, but we're really bad at doing in drug discovery. And that's hitting specific targets, particularly targets that are inside of a cell. So on the left here is a normal neural stem cell, and you can see that it's undergoing division right now, right? Everything's, and this is beautiful. It's kind of the orchestra of cell division. You can see everything is working great there. On this side, you can see there's something really wrong with this cell. This is called a mitotic catastrophe. And the only difference between these two cells is that this is a glioblastoma cell. And in both of these cells, we've knocked down a particular target in the cells. So this cell, we've knocked down this target called BUB1B, and in this cell, we've knocked down this target called BUB1B. And we can knock this out using either siRNA or CRISPR-Cas. You guys have been learning about CRISPR-Cas. So in this experiment, we actually use CRISPR-Cas to completely knock this gene out. You can see it's not essential for the human normal neural stem cell. The cell is still dividing normally, but it basically destroys the cancer cell. This target is not a druggable target, unfortunately. And I'll get into what that means here in a little bit. So just to give you guys a little bit of context about sort of the molecular space that we're working on in Jim's lab. On the right-hand side here, we have antibody drugs. You guys have been learning about antibodies. They're a very, very innovative and cool class of therapeutics. And these are drugs that have basically come to fruition over the last 20 years of research. We really are in what I call the age of antibodies right now. Most of the new biologic drugs that are coming out for things like cancer are antibody drugs. On the far left-hand side here are what we call small molecule drugs. This molecule shown here is Gleevec, and I'll show you an example of Gleevec in a little bit. But small molecule drugs are very small. They're only a few hundred Daltons. These are things you can often take orally as a pill, whereas antibodies are much, much bigger, huge, and these can only be taken systemically, so you have to basically get these injected into your bloodstream for them to work. Most of the drug discovery efforts over the last multiple decades of drug discovery fit into these two camps, either antibodies or small molecule drugs. But there is this area in between that is essentially unexplored from a drug discovery standpoint. Just to go into this and to give you a little bit more of an idea of why we use small molecule drugs and why we use antibody drugs is this example of Gleevec that I'm showing you. So the thing about small molecule drugs is there's only so many targets that you can hit with small molecule drugs. This Gleevec drug binds into the active site of an enzyme. This is the BCR-ABL enzyme that causes chronic myelogenous leukemia. I will tell you, Gleevec is what is considered a miracle drug. This actually cures people of chronic myelogenous leukemia because it I will say specifically binds to the BCR-ABL tyrosine kinase, which is what basically drives this cancer. So this is an example of a very specific small molecule drug. However, there are only so many small molecule drug targets like this, things that we can hit with small molecules. For example, tyrosine kinases as a class are notoriously hard to drug because the drugs are not specific by definition. So even Gleevec hits other tyrosine kinases. And if you look at tyrosine kinase small molecule drugs as a whole, they're very pleiotropic in the fact that they actually hit multiple different tyrosine kinases. So if you were trying to go after one specific drug target, you would actually be hitting all the tyrosine kinases in a human being, which would cause a lot of different side effects. This is the rare example of a very specific small molecule drug. But the most important point to make here is that only about 10% of the drug targets that we want to go after, for things like cancer, for example, are small molecule drug targets, things that we can actually hit. There are only, we like to say, there are only so many enzyme active sites and greasy pockets that we can hit with small molecule drugs. That's a major limitation of small molecule drugs. I will also mention that small molecule drug screening campaigns have been going on for decades in industry. And we're talking, name your target, hundreds of thousands to millions of small molecule drug candidates have been screened against these targets. And it hasn't, even though we've got all this amazing automation and all this ability to screen millions and millions of drugs, we still don't have drugs for the diseases that we're trying to cure. So we sort of hit this limitation in what we can do with small molecule drugs. On the flip side, we have antibody drugs. These are really nice because we can engineer them. We have some really amazing technology to engineer antibody drugs. And we can use these to bind extracellular targets. Antibodies are very big, and basically the only thing we can hit with antibodies are proteins that are on the surface of cells. A really beautiful example of this is shown here. This target here is EGFR. This is the EGFR receptor. And this is the crystal structure of the FAB arm. So there's your FAB arm of your antibody bound to EGFR. This drug is called cetuximab, or Erbitux. It's given to people with colon cancer and a variety of other different types of cancers. And this drug brings in roughly 4 to 5 billion in revenue a year for the drug company that markets it. This is a huge antibody drug, huge antibody drug. Again, though, the point I want to make is that only about another 10% of the druggable targets are accessible by antibodies. That leaves a very good chance that if you understand the biology that's driven the particular type of cancer that you're trying to cure, that you really only have a 20% chance between small molecule drugs and antibody drugs of probably hitting a target that's involved in that biochemical pathway. 80%, the rest of those 80% of drug targets are what we call undruggable. We just don't have the toolbox to actually access those types of drug targets. And that is sort of the impetus of this drug discovery campaign, is that can we focus in on this mid-sized medicine space, this area here, where we haven't done a lot of drug discovery, but where nature has done an amazing amount of drug discovery, and can we begin to start unlocking these undruggable targets? Can we actually design small, mid-sized medicine protein drugs that can get into cells and intercept the types of biochemical pathways that we want to really access with next generation drugs? I said that not a lot of drug discovery has been done here, and in fact it really hasn't. But there are some really beautiful examples that come from natural product drug discovery that fit right into this mid-sized medicine space. And I'm just going to highlight a few examples because I hope it allows you to see sort of where the benefits of this molecular space are. So think antibodies, think small molecules, and then think about these drugs. So the two drugs I'm showing you here are erythromycin and cyclosporin. You can see that they're not small molecule drugs in that they're quite a bit bigger than small molecule drugs. In fact, these things sort of shatter. We have these rules in drug discovery called the Lipinski's rules that sort of talk about what a good small molecule drug is. These drugs do not follow Lipinski's rules at all. These are much larger. They basically break every single Lipinski rule that there is for what makes a good small molecule drug. But these are drugs that were discovered from nature. Back in the day when scientists worked for drug companies and they would go out on vacation, they would literally take soil samples from the places that they would visit. They would bring them back to the lab and they would test for active compounds in those soil samples, like real, true, old school drug discovery. That's how a lot of these drugs were actually found. This is another example of a mid-sized drug. This is vancomycin. And the point I want to make on this slide is that this drug doesn't just fit into a greasy pocket like that Gleevec molecule I showed you. This drug actually grips on to its ligand like a hand. Right? That's a pretty amazing thing that a drug can do. Again, much bigger than any drug that falls into Lipinski's rules over molecular weight of 1,400 Daltons, and it does a really amazing things. Does anybody know what vancomycin is? Brett, you know what it is?
You put me on the spot.
It's a potent antibiotic.
I knew that.
This is kind of the poster child. Cyclosporin is really the poster child for this molecular space, and I really like to talk about it because I hopefully it kind of opens your minds into what we can actually do with this size of molecule. Cyclosporin, again, was discovered from a soil sample. It actually comes from a fungus. And it's a very potent immunosuppressive. If you've ever known anybody that's had to basically go on potent immunosuppressants because of transplant, most likely they're taking cyclosporin. So it's really a revolutionary drug. It basically made transplant possible, this drug. And it does some really amazing things, something that we can't do, we can't design our drugs to do. It actually coordinates this massive 3-protein complex. So you have protein 1, protein 2, protein 3, and here's cyclosporin right in the middle. This drug, as you can see, is not a small molecule. In fact, it looks very peptide-like, right? It's basically a cyclic peptide, although it's made non-ribosomally. But it basically looks like a big cyclic peptide. And it's orally bioavailable, that is, you can take it as a pill. It gets out of your gut, into your bloodstream. It can penetrate into cells and hit an intracellular protein-protein complex. That is sort of the holy grail of drug discovery. Why can't we make drugs that do that? Well, it turns out it's very hard, and it turns out nature has figured this out. So cyclosporin, believe it or not, it actually completely inverts its chemical structure to get into cells. So when it's on the outside of the cell, it sort of binds to the cell membrane, and as it goes through the cell membrane, which has a very hydrophobic interior, it essentially completely inverts its structure, exposing its hydrophobic residues, and then re-inverts itself when it gets into the cell to basically hit its drug target. It's a really beautiful, remarkable example of where nature has figured something out, and we can't even come close to doing this type of design for drug discovery.
Hey Colin, what does cyclosporin do within the fungus that it actually results
That's a
from?
good question. I don't know what its actual what its actual function is, what it's actually evolved to do, but it's a very potent immunosuppressive agent. The issue with natural products, the drugs that basically fit into this mid-sized medicine space, is that they're very hard to optimize their drug-like properties. Pretty much all of the drugs that fit into this space that are in the clinic or that we use to treat patients are the exact drug as they're found in nature. No tweaks have been made to that. For one, trying to tweak something like cyclosporin is very, very difficult. You can see it's a very complex cyclic ring. Trying to synthesize that from scratch at a chemistry bench is very hard. Somebody actually ended up doing it, but it's still made from the fungus itself because you would never synthesize this thing from scratch. So improving or iterating on these drugs is nearly impossible. There are a few of these mid-sized drugs in the clinic. One that's very applicable to the drugs I'm talking to you about today is a drug called Priol that comes from the cone snail. And this is one of these little mini proteins, except it comes from the venom of a cone snail. And it's a very potent analgesic. This is basically injected right into your spinal fluid, and it's used for intractable pain. And it binds to the pain channels in your central nervous system and basically numbs you out. But this is one of these little knotted peptides that comes from the cone snail. It's an example of the type of drugs that I'll be talking about today. I should mention this one just because it's kind of funny. So this is a drug that just hit the market. It's called Linzess, and it's heat-stable enterotoxin is the name of the drug. But it's one of these little knotted peptides that E. coli makes, and it basically causes diarrhea. So they found this molecule, and they basically marketed it as something to treat constipation. So it's kind of funny. These little proteins, these little knotted proteins, as I've been referring to them as, are found throughout nature, believe it or not. Oftentimes they're found in the venoms of spiders and scorpions and snakes and cone snails. They're also found in the leaves of plants. A lot of plants make knotted peptides. In the case of plants, they make these peptides generally for defense. So they basically prevent bugs from eating their leaves by spitting proteases on them that kind of break them down. So a lot of these are actually protease inhibitors. This molecule here is called brazane, and it's found in an African fruit. And this fruit is notorious in African culture because I forget exactly the name that they call it, but it basically is something to the effect of it makes kids forget about their mother's milk because it's so sweet. And this molecule is so sweet because it binds to your sweet taste receptors in your mouth, and it's actually 1,000 times sweeter than sugar. But it's an example of one of these little knotted peptides that we find in nature. Scorpions and spiders, much like the tumor paint molecule that I showed you, make thousands of these things in their venom. And if you think about it, they're really the perfect drug. They're really what we call nature's drugs. They're designed to be injected into whatever organism, their prey, for example, to hit a target with extreme specificity, and to do that and to survive basically the environment of an organism, right? That's exactly, if you think about it on a basic level, what drugs do. right? You get them into people either by injection or by taking them orally, and they need to go to their target and have some sort of a biological effect. That's essentially what these small proteins are. Now, the scorpion doesn't care about curing cancer, obviously, so it's not that these proteins are you're going to inject these into people and they're going to immediately cure cancer. But they have a lot of the molecular properties and drug-like properties that we really care about. And so we leverage this molecular space, and this is where we start our drug discovery processes with these small, mid-sized proteins that we find from nature. Some hallmarks of this class of proteins, these Naughton proteins, they're very small proteins. In fact, I often tell people they're really the smallest polypeptide that actually folds into a defined three-dimensional structure. Typically, they're between 20 and 60 amino acids long. And one of the really cool things is they usually have between 3 and 4 disulfide bridges. You guys know what disulfide bridges are? You've been learning about this in antibody structure, right? Where you have both interchain and interchain disulfide bonds that hold an antibody together. Well, these little protein molecules fold up into very literally knots. That's why we call them Naughtons. And it's classified by this very strict, what we call to and through topology, where you have 2 disulfide bonds that pin the structure together, and then you have a third penetrating disulfide bond that essentially penetrates the other 2 disulfide bonds. It's a very interesting protein topology. It's not something you've probably even seen in your class right now. But because of that, they have a lot of really cool drug-like properties. For one, they're incredibly protease resistant. I'll show you some data where we can throw any number of proteases at these little protein scaffolds, and they're complete rocks. They don't break down, whereas a normal protein would just get chewed up by these proteases. Because they are so small, they're often not immunogenic. And I won't go into the details about this unless somebody is interested in that. More importantly is we can actually engineer these things to have antibody-like affinities. So you don't need this massive antibody structure to specifically bind to a particular target. We can engineer a small protein like this to bind to a target with very similar affinities that you would an antibody. And then, one of the really remarkable things, much like the cyclosporin example that I gave you, is that these little proteins tend to do things that the drugs that we design, things like antibodies, can't. The ability to penetrate through a cell membrane and actually access the interior of a cell, the targets in the interior cell. The ability to cross the blood-brain barrier. The blood-brain barrier excludes about 99% of the drugs that we have. That's small molecule drugs and antibody drugs. Your blood-brain barrier is basically designed to keep things, except for the things that are supposed to go in, out. And it's a major hurdle for drug discovery, especially if you're trying to work on neurodegeneration or brain cancers. The blood-brain barrier is a huge problem. And we have examples of these proteins that are actually able to cross the blood-brain barrier, something you wouldn't predict them to do. Just a note on nomenclature. We call our program the Optide program, which just stands for optimized peptide. So if we take a natural protein from nature, like the tumor paint molecule, and we make an amino acid substitution or we link something to it, it then becomes an optide. Just to differentiate between like a natural Naughton protein and then the drugs that we work with. We call them optides in our group. So these things are found throughout nature, as I mentioned. But it wasn't really appreciated just how abundantly seen they are. And this is one of the first things that we did in the group. So there are a few annotated databases that have these Naughton structures in there. And when we started looking, there was a database called the Naughton database that had about 2,000 different examples of these Naughton proteins from nature. But we went digging bioinformatically, where we basically combed through all the proteomic databases, and we were looking for examples of these knotted proteins. Because they have a variety of different rules that basically specify what their structure is, we're able to use those rules and actually comb through these databases bioinformatically. We identified over 200,000 examples. Now, we haven't annotated these properly, we haven't tested all of these, but we have reason to believe that there's over 200,000 examples of these that are basically found in all swaths of life. So most of the people that have looked at Naughton proteins have focused on a very narrow range of organisms. This is basically spiders and scorpions and cone snails. But these 200,000 actually span all phyla of life, animals, plants. We see evidences of these things actually existing in bacteria, viruses. We don't actually know what their functions are in all cases, but we see examples of what we think are these Naughton proteins throughout nature. And this is kind of like an untapped area where we really don't have any idea what these things are doing, but we'd really like to start diving into these areas and figuring out what they do. Yeah.
Do you think those proteins, it's a case of convergent evolution or divergent evolution?
Yeah, that's a good question. It's hard to tell. In the case of the E. coli STA molecule, for example, there are human ligands that mimic the STA molecule. They're actually signaling molecules in your gut called guanylin and uroguanylin. In E. coli, a bacteria has evolved a function and a structure of that thing that mimics exactly what you see in humans. So there is a possibility where you could see like convergent evolution, but it's not clear exactly what these things are. Like I said, in animals and plants, they're typically either used for predation, so when they have to prey on a particular type of thing, or for protection. I mentioned these drug-like roles, and I'll just kind of go through these pretty quickly, but they're incredibly stable. I'll show you some examples of that. Their ability to penetrate into cells is a little bit repetitive. And then in nature, they tend to hit a particular type of target, which is an ion channel, which is something that we look into a lot in our group, because ion channels are very important for both pain sensation, fluid balance within cells. So that's a very interesting target that we don't actually really hit with small molecules or antibody drugs. So ion channels are something we're very interested in. And then of course protease inhibitors. The issue with this molecular space is that these proteins are very notoriously hard to make. And it's one of the reasons that a lot of drug companies haven't been able to make massive libraries of these like we've done with small molecules, is because they're incredibly hard to synthesize. Most of the time, this is done by actually getting them from the source. And so there are labs, phenomics labs, who have been studying molecules like this for many years, and they essentially do this by milking spiders and scorpions and snakes, and then fractionating the venom, and taking the different fractions from that venom and trying to see what they actually do. Obviously, we're not going to do this in the lab. At least I'm not going to do this. Milking spiders and scorpions doesn't sound fun to me. So what other labs have done is they basically synthesize these from scratch, basically at a chemistry bench, where you link all the different amino acids together, and you synthesize these one at a time. The problem with this is that you would never be able to get through even 100 of these things if you were just synthesizing each one after the other, after the other, after the other. Can anybody think of a reason why it would be tough to just simply synthesize one of these things? Yeah?
Maybe if there's a lot of packaging involved with them, when there are a lot of like later modifications that are done because of the package, they are more tightly.
Yeah, that's exactly right. So the main problem with simply linking all these amino acids together is that you actually have to get the protein to fold up properly. And you saw how it's got all these disulfide bonds that basically determine the three-dimensional structure of this particular protein. You have to get that right. And to do that on this scale is very difficult. Brett has done this many times in his career, where we actually refold the protein. So you essentially make it as an inclusion body, in the case of E. coli. You completely denature it, so it's just a linear string of amino acids, and then you attempt to refold that protein into its actual three-dimensional structure, and you do this in a chemical way where you basically try to get these things to refold. The issue with these small little proteins is they're notoriously hard to refold. So it's a very time-consuming and very expensive process to actually synthesize these, refold them, make sure that you're purifying out the properly folded species, and then move forward with them. So what we did in the lab is actually figure out a way to utilize molecular biology. Can we basically go through all these sequences from nature, clone them out because we understand what their DNA sequence is, and express these recombinantly. And so we built a whole lab that does this. And this is our Optide mammalian expression platform, and this is based on some work that I did as a graduate student. But we have a whole lab on the 5th floor of the Fred Hutch that's basically designed to make these small knotted proteins from nature. And we do this in a mammalian cell line, because using an organism like bacteria, have you guys used bacteria to express a protein in lab? So bacteria are very good at expressing certain types of proteins. However, there's a whole class of proteins, typically proteins that are secreted out of cells, that are notoriously hard to make in E. coli because of this folding issue. You basically have to refold them. So we use a mammalian cell line. This is actually a human cell line to manufacture these little proteins. Mammalian cells have secretory pathways. You guys are familiar with how proteins are basically made in a secretory pathway? A little bit, maybe you've heard of this? Yeah, so mammalian cells are much more sophisticated than E. coli because they have a dedicated pathway with all of the protein machinery that's especially tuned to making proteins like this. Chaperones that help fold it properly, protein disulfide isomerases that help make those disulfide bonds form properly. Basically, it's a kind of a checkpoint mechanism for proteins to get secreted and properly folded out of a cell. It's also a place where a lot of different post-translational modifications happen. Things like adding sugar residues to proteins. These things don't happen in E. coli. They're kind of simple. So we've built this platform to basically manufacture these proteins based on this mammalian expression technology. And now, in basically one production run, we can make about 40 milligrams of any one of these peptides in about 2 weeks of time. The other thing that we've really done, and I didn't have time to throw pictures of this in here, is we've miniaturized this process. So now we have an automated platform at the Fred Hutch that does basically all of this work at a very, very small scale. So this is going to allow us to basically scan through that list of 200,000 sequences that we're really interested in at a small scale and do it very, very rapidly. One of the things that we do when we express these proteins as the mammalian cells, we have to make sure that we biochemically characterize these proteins. And one of the techniques we use in the lab is called HPLC, or reverse phase liquid chromatography. Is anybody familiar with HPLC, or have you used it in chemistry lab before? Yeah, it's a very beautiful technique where we can actually, because of how small these proteins are and how rigid and stable they are, we can almost treat them like small molecules. And so that's one of the reasons that we use HPLC. What I'm showing you here is just a few examples of some of these proteins that we've expressed from nature. And in blue is the trace under non-reducing conditions. That's the normal conditions that the protein exists in. And then in red is under reducing conditions. So we've had a potent reducing agent. Have you guys used reducing agents in the lab?
Which one, guys?
BME?
No.
DTT?
Yes,
Yeah.
right? DTT, right?
So this experiment was actually done with DTT as the reducing agent. And what you can see here is that there's a migration shift on the HPLC chromatogram. And what this tells us is that we have a single species under non-reducing conditions, and then when we add DTT and we break those disulfide bonds that pin this particular structure together, that it unfolds the protein, and its retention time on this reverse phase column changes. This is very critical to us because it's very important that before we take any one of these protein candidates into further drug studies, that we know exactly what we have coming out of the cells. Because we don't know a priori if we're going to make a particular protein, if it's going to come out of the cell and be properly folded. Sometimes this is just a mountain of peaks and it looks like a mess. And we just leave it behind if that's the case. So everything that we make is properly biophysically characterized. You can see this is just a few examples of proteins where we get a single peak as the product and that shifts upon reduction. And as we've continued to do this, we've now marched through and done hundreds of these proteins from nature. We start to see some really interesting things. So this is just a whole bunch of data from a lot of different production runs that we've done in the group. And you'll notice that some of these don't look like the others. So here's one where the red and blue traces are right on top of each other. And here's another one. And these are examples of scaffolds that are actually resistant to reduction. So we can actually add as much reducing agent as we want, and these things are so stable that they stay properly folded. The rest of these all shift upon reduction, and we've now identified a few of these that are so stable they don't even reduce. They completely maintain their three-dimensional structure in the presence of reducing agent. I threw in some really cool pictures. These proteins are also, so stable that we can boil them to purify them. So this is an example of where we actually tried this experiment. We jokingly called this preparative protein poaching. But in this case, what you see here is when we originally express these, we express these fused to another protein. And then we cleave off that kind of chaperone protein, and then we have to purify our little mini peptide away from that big protein. And so one way we hypothesized doing this was to simply just boil the whole milieu. And that's what we did here. You can see that's the first step. And then we pelleted all this down. So this is the fusion protein. And when we take this and run it out on an SDS page gel, this is the protein that's remaining. So we boiled the thing and it's still in perfect shape. And then this is the pellet that you're seeing here, and you can see that this is basically all the crap in the pellet. So this is actually a way that we can purify these proteins, is simply boiling them. They're still completely happy. They don't denature at all. As I mentioned, they're very uniquely protease resistant. So this is an experiment where we're actually testing to see how orally bioavailable these proteins could potentially be. As you know, any drug that you take needs to go into your stomach and basically survive the gastric environment of your stomach, very low pH, a lot of proteases that just non-specifically chew up everything. Basically, if you eat a protein, it gets broken down into amino acids in your gut. That's what your gut is designed to do. So we decided to do a really simple experiment and see how protease resistant are these little protein scaffolds. And so this is an example of one of these proteins where what we do is we first incubate it in simulated gastric fluid. This is basically pH 1 gut juice. And this is the corresponding HPLC trace. And you can see that it's still beautiful, one peak It's not getting broken down by the simulated gastric fluid. We then add 500 units of pepsin. Pepsin is a non-specific protease that only works at low pH in your gut. It basically chews up anything, and it doesn't touch the peptide at all. So this is just a really beautiful example. As a control, we synthesize this peptide without the disulfide bonds. So now, as opposed to this thing being folded up and having a distinct three-dimensional structure, it's now just the primary amino acid sequence. It presumably doesn't fold up into this really tight, well-defined three-dimensional structure. And indeed, when you incubate this is the blue line with 500 units of pepsin and simulated gastric fluid. You can see that peptide just gets completely broken up. In red is the trace of the peptide alone, not in the simulated gastric juice. So you can see that this three-dimensional structure that defines this Naughton class is really, really special, and that it really gives these things a lot of great drug-like properties. So this is an example of a protein, for example, that you could potentially take orally, and it could get into the bloodstream. You guys have been working with a lot of crystal structures in the lab, looking at different three-dimensional structures in Pymol. You guys been doing that? Yeah. So this is a crystal structure of one of these Naughton proteins that we solved in our group. And nobody has really studied these three-dimensionally. A few groups have, and they use a technique called NMR to do that. But we've actually solved the crystal structures of these, again, making sure that what we're making is exactly what we think we have. And in this case, I'm showing the electron density for the 4 disulfide bonds that basically staple this particular protein scaffold together. And you can see that the disulfide bonds are exactly where they should be. So again, this is just another really beautiful example of how we can express these proteins from nature unlike anybody else in the world. We can characterize them both biochemically and using structural biology. And that gives us a lot of tools for drug discovery. Yeah?
Are those disulfide bonds consecutive within the primary amino acid sequence? Like does cysteine 1 disulfide bond to cysteine 2?
Nope, not at all. So typically, depending on the type of Naughton you're talking about, they'll have very specific disulfide connectivities. So it'll be like a 1, 3, 4, 5, 7, 8 connectivity. It really depends on what three-dimensional scaffold you're working at. These things don't just look like this protein. They come in all different shapes and all different sizes. So the connectivity can be very different.
So when these things are folding as they're synthesized, probably the entire polypeptide synthesized and then it's folding? Like in other words, it's not folding as it comes off ribosome.
It's hard to imagine that that's what's happening. Most of these peptides, as they're expressed in nature, they're expressed in venom glands and they're expressed as secreted proteins. So they have a signal peptide. I don't know if you guys have learned about signal peptides, but they have a leader peptide that basically tells the cell this thing needs to be secreted. And so as it's getting translated into the endoplasmic reticulum, it's probably not the case where it's just folding as it's getting translocated. It probably gets shuttled into the ER and then kind of binds to different chaperones or protein disulfide isomerases that help it fold properly. We don't really know. People haven't really studied the folding kinetics of these things. Some people think they just automatically just snap together, and the disulfides have to come together exactly the right way. And if they don't, you sort of have a mess.
Well, so you guys mutated, when you guys made the mutant, were those cysteine to serine?
Cysteine to serine mutations.
And that thing spontaneously, that thing folds up, right? You saw that thing at the gastric, you had that nice peak.
Well, it doesn't necessarily mean that it's folding properly,
Okay.
but it definitely means that it's some sort of a species.
Okay.
Yeah. Most likely it's probably an unfolded peptide, but we don't really know. We weren't able to characterize it structurally. One of the beautiful things about being able to structurally characterize these proteins is that we can use that information as the basis for computational design. I don't know if you guys have learned much about the powers of computational design, but there are people, especially in Seattle, who are very good at basically making designer proteins on a computer and then testing them. And so one of the things that we can do is characterize these proteins three-dimensionally, and then we can feed them into computer programs and actually design our protein. This is an example of an actual design in our group, where we take a Naughton scaffold, here shown at the top, and we actually use the computer to engineer in a binding interface. So again, we're basically trying to engineer an antibody-like affinity into this particular cross-linked scaffold. And you can see here, what the computer program is trying to do is make mutations in the Naughton scaffold that will basically make it stick to the target of interest. Right? So this is engineering a protein-protein interaction using one of these Naughton proteins. And so This target that we're actually designing against is an intracellular target. This target exists inside of cells. It's an undruggable target, meaning we don't have small molecule drugs against it. Because it's inside the cell, we can't access it with antibody drugs, but we can now design one of these small knotted protein drugs that could potentially penetrate into the cell and access this particular type of target. So from start to finish, we can mine these sequences, we can express them in the lab, we can characterize them biochemically and structurally. We can take all of that information, feed it into design algorithms, and we can begin making designer drugs based on these drug-like scaffolds from nature that are incredibly rigid, protease resistant, essentially everything that you want in a good protein-based drug.
Hey, Colin.
Yeah.
So in between, so the loops that are in between
The cysteines. Yeah.
In nature, is there a variability with how big those loops are, or can you engineer those loops?
Yeah, oftentimes there is. So he brings up a really good point. For example, if you take one of these proteins and you align it to a whole bunch of its related family members, you can see where nature has made mutations. Nature has sampled different mutations within the scaffolds. In some cases, Brett, those loops are extended much longer. In some cases, they're truncated. So his point is very well made. Outside of that cysteine scaffold that basically locks this thing together, all of those loops are engineerable. So that's actually something we do often in the lab. We'll take out the native loop and we'll replace it with a loop that we're interested in. So they're highly malleable, actually, in that case. But nature actually guides us for a lot of things. It kind of tells us where we can make mutations and where we can't. And we use that information to inform our design process a lot. I should also just mention here, so I'm sort of telling you about the fact that we've made 100 of these things and that we've miniaturized this, and so we can maybe make a few 100,000 of these things. But the reality of drug discovery is that, statistically speaking, you need much larger numbers to hope to get new things. So for example, if we were hoping to design something against a target on a computer, you're not just going to design it and that one molecule is going to work. The reality of the situation is you need to make hundreds of thousands to millions of variants of that particular design to get one that's really going to work well. This is basically a sample size issue, right? We need to work with millions, maybe even billions of these things to find the ones that work the best. And so to do that, we've actually designed a system where we can take these little knotted peptides and we can actually link them to the surface of a cell, in this case a yeast cell or a mammalian cell, and we can make very large display libraries. You guys probably haven't talked about this in your lab yet, but this is just a way to basically make hundreds of thousands to millions of variants of a particular scaffold and screen through them on the surface of cells very quickly using techniques like flow cytometry. So this allows us to basically take very interesting leads and make many, many variants of them and pick out the best ones. One of the really cool aspects of our program is that we've kind of followed this tumor paint molecule, and that, listen, if we're going to make a drug, if we're going to put something into the clinic as quickly as possible, we need to get that preclinical data as quickly as possible. So we basically do all of our work. We do all the biochemistry work, but the next step is we go directly into animal models that have either the type of cancer or the type of disease that we're trying to study. And so I'm going to show you some really cool images of where we've done a very simple experiment. We've taken these proteins from nature, we've labeled them with a radioactive tracer, and we've asked a very simple question. If we put these into an animal, where do they go? Can nature tell us something here about these proteins? And we've seen some really interesting things, and I'll go through this kind of quickly. These are examples of some of these proteins that go to the kidneys, and most of these proteins actually get washed away through the kidney because they're so small. As soon as you inject them into the bloodstream, they basically get excreted in your urine, and so their natural path is through the kidney. However, we found proteins that bind very specifically to the proximal tubules of the kidney. The proximal tubule is the area where most protein recycling happens in the kidney. This is a very interesting thing that we never expected to find. Most of these proteins just get flushed right through the kidney very rapidly, and we can see that. But these ones hang around for many, many days post-injection. That could be a liability in the case where you're thinking of this thing as attached to a cytotoxic drug. That could be very toxic to the kidney, but it could be very useful in treating certain types of kidney diseases. Most kidney diseases originate from damage to the proximal tubules. We have examples of these proteins that penetrate the blood-brain barrier. This is a very remarkable phenomenon and something that we would never have ever predicted from first principles. Remember what I said? 99% of the small molecule drugs and protein drugs we have do not cross the blood-brain barrier. we found family members, classes of these molecules that cross the blood-brain barrier. We have a very active project in trying to figure out how we can leverage this property to bring things like antibody drugs across the blood-brain barrier or small molecule drugs across the blood-brain barrier so that we can begin to do things like treat brain cancers or neurodegeneration diseases like Alzheimer's. We also are really interested in this tumor homing phenomenon. We have a bunch of different molecules that we've identified that have similar properties to chlorotoxin. That is, we can take off the near-infrared dye from chlorotoxin. And this is an example of this where we have the naked peptide one, where we've added a radio tracer. And then we've got basically the tumor paint version of this peptide where we've radio labeled it. And you can see that in this case, for this particular peptide, that even if we take the dye off, it still homes to the tumor really, really well. So we have a very active program, big active program right now where we're basically trying to make, Django talked about antibody drug conjugates the other week. We're trying to make peptide drug conjugates. Can we leverage the tumor homing properties of these peptides to deliver cytotoxic agents into the cancer? And then we found some really cool things that we never thought we were ever going to find. And I'm going to show you an image of when we saw this roll off the imager, we were like, what the hell is going on here? This is an example where we took one of these natural peptides from nature, again, radio labeled it, and administered it to the mouse. You notice something very strange? The peptide, which is all these dark spots, is localizing very, very specifically to the cartilage of this mouse. We're not exactly sure what to do with this yet. As you can imagine, we're very interested in things like osteoarthritis. These are huge drug markets, as you can imagine. But we have a peptide that we've isolated from nature, well, using molecular biology techniques, and we radio labeled it, we put it in the mouse, we asked where does it go, and remarkably this peptide has some sort of remarkable homing property, has some sort of zip code within the mouse that it knows how to home to cartilage. This is something we never would have thought of. This is an example of sort of, in a sense, the way we used to do drug discovery, which is more phenotypically. Take drugs from nature, put them on things, and see what they do. And this is a remarkable example of something that we've discovered in this molecular space. So hopefully that gives you a bit of an idea of some of the stuff we're working on in Jim's lab and where we're trying to take this tumor paint story. And we're done at 1:30 or?
We get to 1:35.
Oh, okay, perfect. And so I just sort of wanted to end this talk by talking a little bit about just being an innovative thinker. And as you go through your training, you guys are still young in your academic careers, and depending on whether you go into science or medicine or whatever it is, I just want to encourage you guys to really think innovatively and to not necessarily take the path that's just laid out for you, to think about ways that you can kind of go against the grain and do things differently. Jim is a really remarkable example of somebody that has done this. He is a classically trained physician. He takes care of kids with brain cancers day in and day out. He prescribes them the same drugs day in and day out, and they have no effect. And he's been doing this for 20, 30 years of his career, and we've been doing this longer in the course of drug discovery, where we continue to do the same things over and over again for lack of anything better. And so another example of something that came out of our lab is called the needle array device. And this is a company that he spun out of the lab called Presage. And what they did is commercialize a needle array device that's shown here in this, that got the cover of Science Translational Medicine, which was very cool. And he did a very simple thing, much like the tumor paint molecule. He said, listen, why are we blindly giving patients drugs that we know are not going to work? Why don't we at least develop something where we can screen drugs in a patient beforehand and at least have a slight idea of what drug might be effective against that particular type of tumor? So the way the needle array device works is that they put multiple different chemotherapeutic agents into each of the needles of this particular pen. It's basically about the size as a handheld device. Before surgery, this pen is injected into the tumor, and all of the drugs are basically injected into the tumor, leaving a column of each drug throughout the mass of the tumor. And then when that tumor is resected and we send it down to pathology, let's ask ourselves which drug is working, which drug is killing those cancer cells before we actually give that drug to the patient. Very simple, right? Theoretically in your head. I mean, this makes so much sense. Why haven't we done this? This is an example of the sort of the thinking that we do in the lab and the thinking that Jim encourages us to do in the lab, which is, listen, just because no one has done this before, or it may seem really, really stupid, let's try and do this. And the needle array device is a really cool example of something that has spun out of this thing that's like, let's just do this. Let's just see which drugs work well in the tumor and which don't. And let's maybe use that to inform decisions. Or maybe let's take all these drug candidates that are sitting in drug companies all over the world that have basically gone nowhere, and let's start combining them together and seeing maybe drug 1 and drug 2 don't work individually, but they work synergistically. And let's use a technique like this to start answering these types of questions. That's the basis of Presage, which spun out of Fred Hutch probably about 6 or 7 years ago. I also want to leave you with one other point, in that it is damn hard to do drug discovery, and it is damn expensive to do drug discovery. This is from an article in Forbes magazine that was titled "The Truly Staggering Cost of Drug Discovery." And it's a little bit weird how you come up with a number like this, and there's a lot of debate on what the right way is to come up with a number like this. But essentially what they did is they sort of looked at all these drug companies, they took the successes and the failures, and they said, taking into account the cost of failure, what does it take a drug company to develop a drug? And look at these numbers. The best company, Amgen, cost them $4 billion per drug. Companies that are not doing so good, sell the stock if you own it, just kidding. AstraZeneca, $12 billion per approved drug. We have a big problem right now in drug discovery, and that is actually becoming economically unviable to do R&D work to develop drugs. How can it be economically viable when it costs $12 billion for an approved drug? We have a bit of a crunch going on here, and what we're seeing in the industry is that the big pharma companies they're stopping R&D because it's not working, and they're essentially going out and trying to acquire innovation from smaller companies. This is creating a really cool environment for young scientists to do this type of science. And this type of science can be very, very lucrative, of course, and you can actually have a big impact on people's lives. But it's really creating a new opportunity for kids like you who are coming up and getting trained in science to be doing this type of stuff. You don't actually have to go work for the Amgen or the AstraZeneca to do drug discovery. This can be done on a much smaller scale, and these companies will help fund this research because they have so much money to do this. The other really striking thing is we haven't gotten any better at doing drug discovery. We've become incredibly efficient. Our brute force techniques for doing drug discovery, things like making antibody libraries, screening small molecule drugs, our brute force efficiencies have gone up hundreds, thousands fold from back in the day when we used to drug discovery by basically taking soil samples out of the soil and bringing them into the lab. Why is that? Why have we had these huge increases in brute force efficiency, yet it's more expensive, we have more failures in the clinic, and we're just overall almost worse at doing drug discovery right now? I don't know the answer to this. It's just something I want to pose to you and as you guys think about things like drug discovery, because it's a big problem and people have thought about this a lot, and there's a lot of decision theory analysis that goes into this. It's like, well, how is this possible that we have these increases in efficiency, yet we're getting worse and worse at developing new drugs? A lot of that is we're trying to go after really difficult things that are a lot harder than even nowadays with our technologies than the things that we've gone after before. That is, the low-hanging fruit has been harvested. But a lot of that has to do with just the way we think about drug discovery and the way that we think about target-centric drug discovery. That is, we're so interested in what is this target, is it validated, can we go after it? Every single drug company is going after the exact same target, spending all these billions and billions of dollars to go after the exact same targets because they think they're the right targets, as opposed to maybe going back to the beginning a little bit and saying, hey, are we going about this the right way? Are we studying the right models? For example, are cancer cells in a dish a good way to discover drugs? Are cancer cells in an animal a good way to discover drugs? There's just all these questions, and I don't want to leave you on too much of an open-ended discussion here, but the point is philosophically we have a lot of problems in the way that we do drug discovery now, and so I want you guys to sort of always be thinking outside the box when it comes to whatever it is that you're doing, because we have almost a crisis in the drug discovery industry right now, and it's something that our lab is trying to address. So, thank you. I ran out of time. I can talk about this all day, of course. If you guys have questions or you want to come by and see the lab, make sure you talk to Dr. Kaiser. We'd love to have students in the lab. Did you take this group through the lab?
Allison was one of the part of my group
Okay.
that we went through in the winter quarter. You guys did protein projects. We took a field trip down there. It was awesome.
So thank you guys.
All right, let's thank Dr. Correnti.